Hi, so we're continuing on our discussion of enzymes. Last time we stopped here at NADPH, so we're going to continue talking about that. Or we stopped with NADH, sorry. Now we're going to talk about NADPH. So that works the same way. Notice that they have this kind of like in parentheses here. So basically it's going to act like a hydride source. And that's really it's a really important feature, but that's its feature. So it's going to act like a hydride source, and so it's a reducing agent. So here we have folic acid can go through dihydrofolate reductase to dihydrofolate, and we can have it go to tetrahydrofolate. And this is also tetrahydrofolate. It's really useful because it can transfer 1 carbon units. And so this is really important, you can imagine, for both biosynthesis and catabolism, so breaking down of products as well. So another set of coenzymes that's really important are the flavin coenzymes. So that's from the Latin word for yellow, flavus, and that these pigments give both the enzymes that are yellow themselves, and also when they're coenzymes, they give the enzymes that they're associated with a yellow color as well. And so there on the right, you see that flavin structure, the core, and then you see 2 different structures. We see FMN, which is the flavin mononucleotide, and then we see a flavin adenine dinucleotide. So that structure on the bottom there is what we're going to call a nucleotide, that aromatic, the 3-ring system. And then when we have over here on the far right, we have a DNA base adenine, and that's coupled to the flavin. So there's 2 nucleotides there. So flavin is really important because it can do redox chemistry, it can do reduction and oxidation chemistry, and it can do 1 electron versions of this. So that's really important. So what happens is that overall, the reaction is 2 electrons, right, to get to the fully oxidized and the fully reduced, but you have this semiquinone form. And this semiquinone form is this radical anion, and this is a 1 electron transfer in between the 2. And so this is a way that your body can safely do radical chemistry without it being destructive to the cells. And so the flavin chemistry is super fascinating. We could probably do a whole course basically on flavin chemistry and all the ways it works in the enzymes. Don't worry, that's not this course. But I'll just tell you that it's really cool. And these enzymes that have this flavin cofactor, so again, this is something that you need to eat with your food. It's a vitamin. Your body can't make it. Enzymes that use this are called, as a 1 electron receptor or acceptor, are called dehydrogenases or dehydrogenases. And they work by a number of different mechanisms, so it's pretty complex. So this is, again, that's what I was saying before, is that we could teach a whole class on this. So I'm going to go through this a little bit quickly. But here's an example of monoamine oxidase. So this is important because this is how drugs, or not just drugs, anything in your body that has an amine, and this includes things like dopamine and serotonin, how they are broken down and destroyed, basically, because your body biosynthesizes things and it also destroys things. And so this is how MAOIs, monoamine oxidase inhibitors, enzyme inhibitors, which we'll talk more about next class, work by inhibiting enzymes that do this reaction. So they're going to get in here and mess with this radical chemistry somehow, or mess with the binding of the substrate so that it can't do this radical chemistry, because this is how something like dopamine that has a primary amine is going to get turned into an aldehyde. So it's pretty important chemistry. And notice we're generating peroxide there. It's crazy that this isn't more destructive to the body than it is. So flavin monooxygenases, they're important in drug metabolism in general, in addition to neurotransmitter metabolism and any kind of amino acid metabolism, except for proline. And they do a number of different oxidation chemistry steps, which is really interesting. And notice here that we've got a hydride from NADH, in this case, is doing part of the chemistry. And one of the reasons that it's so important in drug metabolism is it allows you to get one oxygen from O2 into a substrate, so it's flavin monooxygenase. So through the intermediacy of that flavin cofactor there, you could do radical chemistry, because remember, oxygen is a diradical in its ground state, so it's going to want to do radical chemistry. So you do radical chemistry with that oxygen, and it's going to enable one oxygen to be incorporated into the substrate. When oxygens are incorporated into your substrate, this generally will help your substrate become more polar and more water soluble. And so remember, if your drug is really water soluble, it's not going to want to go to its target. And where it ends up going is it stays in the bloodstream and goes to the kidneys and is excreted through the urine. So that's why it's really important, if a drug can get oxidized by a flavin monooxygenase, it is a way that drugs can get metabolized and directed towards excretion. Another important cofactor is the heme cofactor, and so we've talked about this before because this is cytochrome P450. So I've drawn this before as a square, and you can see now why I would. The coral system is big. This is important because, again, it adds hydroxylations and epoxidations through using oxygen insertion chemistry, because iron and oxygen like each other. So we can see here that it's going through, amazingly, it's going through an iron 4 center, and it's able to, through this and also a key sulfur, is able to add oxygens by a radical mechanism to carbons. It can also do epoxidation, which is really neat, and you could see sulfoxidation. This is actually, honestly, I'm less impressed by this reaction. The other two are harder. Okay. ATP and coenzyme A. So we've talked about this before. I think you guys are all familiar with adenosine triphosphate and how it provides energy to molecules to get them to do chemical reactions. So you probably heard of the analogy that it's like the biological currency before. And what ATP does is it provides energy to molecules to get them to react. So let's think an example here is that if you mix benzoic acid with ammonia, you wouldn't get the amide, and that's because, see, the H is not a good leaving group. You need to do something to make things a better leaving group, and actually that's a lot of what ATP-CoA, this cofactor, does. So you have to activate this molecule. So this is ATP, is an activated molecule, and a lot of times ATP-CoA together are going to act like that leaving group. There's other ways, though, that it can work so that you can indirectly break these bonds, and notice that you have these anhydride bonds that can be broken by a nucleophile. So what we do is that we can, let's say that we want to break down a fatty acid or something, we can create an acyl-CoA derivative where it used to be, see, this is what it used to be. And this whole process of chemistry is essentially to turn OH into a good leaving group. So now this whole big giant thing we'll call coenzyme A together. So that's pantothenic acid. Remember, it's another of your B vitamins. And we now have a thioether, a thioester. So it's a mixed ester. And what we're going to do here is that this whole thing is now a good leaving group. So the thioester group, you can have that alpha proton's now more acidic. They're much more reactive to acylation. They can do a bunch of different things, and they're not gonna break down in aqueous media. So they're not one of my super hot electrophiles that I love so much. So briefly, we'll say that you can use enzymatic synthesis. This is switching gears. You can use enzymatic synthesis to make chiral drug intermediates. So this is typically you use these lipases to do different chiral resolutions. So you take a mixture of stereoisomers and the enzyme resolves them into one enantiomer for you. Enzymes are used more and more in catalytic reactions to make drugs. And so here's an example using sitagliptin. And another place that this is really was used was in artemisinin, which is, so it was a semisynthesis to create an anti-malarial drug. People are also given enzymes if they have certain genetic enzyme deficiencies. These can have a lot of issues whenever you're giving people protein as a dose, and that relates to degrading proteins, like we talked about before, the poor bioavailability of proteins, and also people can develop allergies to proteins. Okay, so that's the end of it. So that's the end of lecture 16, and so I'm going to end that, and then I'm going to start the ends of our discussion of lecture 16 slides, and I'm going to start on lecture 18, and so that should be reading chapter 5.